the sound? Okay.
Okay.
Okay. Before we start, I will talk to greet to my participant from Indonesia first with Bahasa Indonesia. Please allow me just maybe a little bit a short minute to explain about the our this lecture today. Oke, selamat pagi. Assalamualaikum warahmatullahi wabarakatuh pada Bapak Ibu semua, para mahasiswa program Magister Ilmu Kedokteran Tropis UGM. Ya, pada kesempatan siang hari ini, pagi hari ini, kita merupakan bagian dari guest lecture series yang ketiga. Ya. Sebelumnya kita mendapatkan insight dari Profesor Tom Burkott dari James Cook University dari Australia. Kemudian selanjutnya kita dapat perkuliahan dari Profesor Sylvie Mengger dari Hydroscience Montpellier dari France. Dan yang ketiga kita pada hari ini bahagia sekali kita mendapatkan tamu spesial Profesor Atiporn Saeung dari Chiang Mai University di Thailand. Okay, good morning everybody. Ladies and gentlemen, especially for Professor Atiporn Saeung from University of Chiang Mai. Very happy to see you again. Yeah, thanks you for being willing to give the lecture today. And of course, esteemed faculty member of Faculty of Medicine, Gajah Mada University, Faculty of Medicine, Public Health and Nursing, Gajah Mada University, and student of Master Program of Tropical Medicine, Gajah Mada University. And all of the distinguished participants, especially from the Ministry of Health of Indonesia, that I see many people from the Ministry of Health also attend here, and National Research and Innovation Agency and other research institute, Indonesian Pest Control Association. Thank you for your coming also, and all the participants that I cannot mention one by one. So good morning and welcome to today's guest lecture organized by Master Program of Tropical Medicine, Faculty of Medicine, Public Health and Nursing, Gajah Mada University. So I'm delighted to introduce our distinguished speaker today, Associate Professor Atiporn Saeung, who will enlighten us on the topic of Anopheles Species Complex identification and implication for vector control. Yeah, before we start the guest lecture, so I would like to little bit read her curriculum vitae. So Associate Professor Dr. Atiporn Saeung is a head of parasitology of Faculty of Medicine at Chiang Mai University in Thailand. She is also Associate Professor in Medical Parasitology and Entomology and also Medical Important Mosquitoes and Black Flies. She is the author of more than 100 index publications and served as a reviewer in many scientific journals. So her PhD obtained from the Department of Parasitology at Chiang Mai University, and we know that she is one of the leading medical entomologists and academic researcher, especially in the interest concern of Anopheles species complex. So please join me in the extending a warm welcoming to Professor Atiporn Saeung. And Professor Atiporn, the floor is yours. Okay.
Thank you so much, Dr. Tribo, for your kind introduction. And good morning, everyone. Nice to meet you all. And first of all, I would like to thank Dr. Tribo and the Faculty of Medicine, Public Health and Nursing UGM for their kind invitation. I'm deeply honored to be invited to speak here today. So I will start my class. I will share my screen.
Can you share the PowerPoint?
Yeah.
Okay.
What's disable screen? This with for the is host disable participant screen sharing.
Okay. Please the committee to open the host for Dr. Atiporn. Could you please to try again, Dr. Atiporn?
Okay, I will share.
Okay. Yeah. Perfect. Yeah.
Okay. Today I'm gonna talk about Anopheles species complexes, identification and implications for vector control. And before we start the class, I'd like to invite you to scan the QR code to participate the activity before we start the lecture. This is the Mentimeter QR code. Okay, and then please type any words or topics that related to Anopheles species complexes that you know about Anopheles species complex. Any words, yeah, please type after you scan the QR code. Okay, I can see your response from my screen. Okay. Okay, yeah, some students. Wow, Anopheles dirus, spring species. Oh, okay. Fabulous trees.
Silahkan share code Bapak Ibu teman-teman untuk kemudian men-type kira-kira Anopheles apa namanya
Yeah. Wow,
spesies
perfect.
kompleks apa saja
Yeah,
ya.
perfect. Yeah, yeah. Wow. Most of all have the basic knowledge of the Anopheles vectors. Yes. Yeah. Some students write cryptic or spring, malaria, vector-borne disease, same morphology. Okay. Some students write down the vectors.
Materinya belum kelihatan ya?
Belum, Mbak Andi, baru scan code ya. Ya. Apakah kelihatan scan code-nya?
Masih yang slide pertama?
Wow,
Ya.
now
Ya,
42
kelihatan, Pak.
response. Yeah. Okay, falciparum, ovary, some students write down the malaria parasites. Oh, geometric morphometrics. Wow. Oh, Anopheles Sulawesi.
Yes, we have also Anopheles Sulawesi.
Sulawesi.
Yeah.
How about the vector of malaria in Indonesia?
It's about 29 species of Anopheles that confirmed as a vector in Indonesia.
Oh,
So we have lots. Yes, 29 and 30, yes.
Oh,
So, and a part of this is Anopheles species complex. So this is very important to know about it, how
oh,
to identify the species complex and what the implication of the vector formula.
that's fine. So, how about Anopheles barbarostris? Barbarostris is
Yes.
the primary vector, right?
Yes,
In
of course, in Sulawesi and also in East Nusa Tenggara, the Barbarostris, Anopheles barbarostris is a
oh,
main vector of the malaria and also lymphatic filariasis.
versus wow, and for Anopheles dirust,
Actually, we suspected some species of the Leucospirus group that have a morphology as Anopheles dirus, but we need to reconfirm it
oh,
with our colleagues, maybe especially from your lab and also from the Malaysia colleagues, so to make sure the species is Anopheles dirus. Yeah.
okay. I think now 58 response from the audience. Yeah, thank you very much for joining the activity. Yes, but I know, yeah, some most have like a knowledge on Anopheles species complex. Yeah, thank you very much for joining the activity. Okay, thank you so much. And เดี๋ยวไปต่อเลยเนาะโอเคแป้งแล้วพี่จะติดตรงนี้ได้ไงเอาไว้ข้างล่างเลื่อนลงมาได้ไหม excuse me okay yeah
Okay. Go ahead.
okay yeah okay, thank you okay as you know that the malaria is one of the most important mosquito-borne disease caused by Plasmodium parasites transmitted to humans by infected Anopheles female mosquitoes. And for humans, malaria species includes, as you some audience already tied, Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, and Plasmodium ovale. And species of the leucospirus group, for example, Anopheles dirus complex and Anopheles barbarostris. The minimus subgroup that are Anopheles minimus and Anopheles volatilis sensulato, and the maculatus group that are Anopheles maculatus, Anopheles pseudovimoli, Anopheles swattwongpornai, and Anopheles wilmoli are the primary or secondary vectors in rural and forest areas across Asian countries. And species of Anopheles culicifacis and Anopheles sapitas complexes are the primary vectors in India and Sri Lanka. Anopheles sandecas complex is known to be a vector of the important vector along the coastal areas and islands in South and Southeast Asia. And Egyptites and Abopictus are the vectors of dengue, chikungunya, and Zika virus infections. And Culex species are the vectors of Japanese encephalitis. And why is the dengue vector so difficult to control? Yeah. Anybody can tell me some reasons why is dengue fever so difficult to control? Yeah. I'd like to know the opinion from the audience. Yeah. could representative person tell me some reasons why is dengue fever so difficult to control?
Maybe any person that who will answer the question from the Dr. Atiporn, please raise your hand or maybe go ahead directly.
Yeah.
This.
Yeah, please, no correct or incorrect. Yeah, just or your opinions. Yeah, from your opinions, why dengue fever so difficult to control? In your opinion. Yeah, please feel free to answer.
Yeah, someone answer from the chat room, I think, Dr. Atiporn. Yeah. Oh, Sidik. Oh, yeah.
Okay.
Can you see in the chat room, Dr. Atiporn?
Yeah,
Yeah.
or chat room. Chat
Yeah,
room.
chat room. Yeah.
Chat
Some
room.
people, some participants answer the question from the chat room.
Oh, okay. Sorry, I will ask my staff. I cannot see the chat. Excuse me, please wait for a while.
Okay. Mohon break sebentar ya. Ya, terima kasih.
In the chat. นี่ก็เปิดแชทไว้ได้ค่ะ
So maybe if you cannot see the chat, I will.
เดี๋ยวแชทมันจะมาเรื่อยๆนะครับ
พี่ก็เปิดตรงนี้ได้ ก็คือเปิดดูจากตรงนี้ใช่ไหมคะ
ครับเนี่ยมันจะมาเรื่อย
แล้วพี่ก็เปิดตรงนี้ เวลาปิดก็ปิดตรงนี้ใช่ไหม
ๆผมเปิดทิ้งไว้นะครับ
โอเค exploring knowledge
เดี๋ยวเพื่อนฟังดีนะครับ
because there are many difficulties to conduct effective vector control. Yes, correct. And dengue outbreaks are influenced by environmental factors like temperature, rainfall, humidity, which affect mosquito breeding and survival. Oh, good job. Some more question, some more answer. Okay. ที่ไหนก็ปิดตรงนี้เลยใช่ไหมหรือว่า
ทิ้งไว้เลยก็ได้ครับ
เปิด
เปิดทิ้งไว้นะครับ
ไม่ต้องนั่นใช่ไหมคะ
ครับแล้วก็
แล้วมันไม่ปิดเองหรือว่าเราเอาไว้ย่อไว้ข้างล่างเลย
มันจะหายนะครับ
มันจะหายไงพี่แบงค์เปิดไว้ออกไว้ตรงนี้ให้ได้หน่อยโอเค Thank you. Okay, yes. And the answer that you wrote from the chat is correct. And for the control of dengue transmission is difficult for several reasons because no effective dengue vaccines is available for use in the community, and there is no specific drug for treating dengue infection. And the mosquito of Aedes aegypti vector has developed the resistance to several classes of insecticides used as larvicides or adulticides. So it hence reduces the efficacy of vector control using insecticide-based methods. And other reasons are the people usually collect the water in containers in their properties to provide breeding sites for the vectors. And the important reasons, the virus can pass from the female to their eggs, or we call vertical transmission. The emerging females can readily be infected without taking any infected blood meal. The male mosquito that emerge from infected eggs can also be infected and transfer the virus to virgin female via the copulation. This can maintain the virus in the vector populations in the absence of human and other vertebrate carriers. These are some reasons that's why the dengue fever is so difficult to control. For the vectors of lymphatic filariasis, Q. fasciatus are the major vector of Wucherenkopfstein across urban and semi-urban areas in Africa, Asia, and South America. And some species of Aedes and Anopheles are vectors of Wucherenkopfstein in rural and forest areas. Yeah. And also some species of Mansonia and Cocuritidea are vectors of Bukia malaria. And some species of Aedes are the vectors of Wucherenkopfstein in endemic islands in the Pacific. And Brucia timori is confined to only the Lesser Sunda Islands of Indonesia, right? Brucia timori.
Yes,
Yeah.
Exactly. In a Lesser Sunda, yes.
Lesser Sunda Islands. Why the correct identification of mosquito is fundamentally important for studying mosquito-borne disease? Because not all Anopheles species are vectors of malaria parasites, and different species of Anopheles vector have different vectorial capacity or biting behavior or bionomics and insecticide resistant status. Anopheles species being vector in one place may not be vector in another place, and no all-in-one approach to be effective for controlling all malaria vectors and stop malaria transmission. How about the problems and challenge for identifying the Anopheles mosquito species? Because most identification keys are for females because of their medical importance, but females of many important genera, for example, Anopheles, Culex, and Ocelotetus, cannot be distinguished. What are the cryptic and isomorphic species? Nearly all malaria-transmitting Anopheles studies to date have proven to be groups of sibling or cryptic species. And females of many species are extremely similar, or we call isomorphic, and cannot be distinguished reliably. The challenge is to accurately identify these species so their bionomics and relation to disease can be studied. The elucidation of isomorphic and sibling species require the use of morphological or classic taxonomy and molecular or DNA techniques. However, some genetic techniques like cytogenetics and electrophoresis are rarely used today. Let's see the definition of cryptic species complexes. Species complex is a group of closely related organisms that are very similar in appearance to the point that the boundaries between them are often unclear. For cryptic species or sibling species, is one of two or more morphologically indistinguishable species that are incapable of interbreeding. Remarkably, the malaria control personnel in central Vietnam were applying residual insecticide inside house to eliminate the suspected malaria vectors. A morphological variant of Anopheles minimus, and the malaria case rate was not affected. Subsequently, the comparative morphology and later DNA PCR showed that the morphological variance was Anopheles varuna, which is a non-vector. And the result of misdiagnosis was a costly waste of resources. The control method was not effective against the actual vector, which is Anopheles diarrhea. So that's why we have to correctly identify species of Anopheles species complex. And here is the summarized finding of mosquito species in the world. Nowadays, a total of 3,726 species of mosquito are currently formally recognized. Mosquitoes are organisms classified in the phylum Arthropoda, class Insecta, order Diptera, suborder Nematosura, and family Corycidae. This family is classified into two subfamilies, Anophelinae and Corycinae. For the classification of mosquito species, we use genus and subgenus, and use species and subspecies that are regulated by the International Code of Zoological Nomenclature. But for section, series, group, subgroup, or complex are not regulated. And Anophelinae, subfamily Anophelinae, are divided into three genera. Genus Anopheles, 8 subgenera, genus Byronella, 3 subgenera, and genus Trichasia. And based on a classification of the triad Edeni proposed by Leonard and others, the subfamily Corycinae has 110 genera. However, Wilkerson and Arthur in 2015 argued that Reinert classification of Edeni may cause confusion and is not applicable for many reasons. To stabilize the ED classification and maximize its usefulness to the population community, the authors encouraged using the traditional classification and reduced the new genera proposed by Reinert to the subgeneric level. And now we follow the traditional classification suggested by Wilkerson and other in 2015. As a result, the number of genera in subfamily Corycini is reduced to 38. And here's an example of mosquito classifications. Anopheles babylotris belong to subgenus Anopheles, Laticon section, Myxorhyncha series, Babylostid group, and Babylostid subgroup, and Babylostid complex. For Anopheles minimus belong to subgenus Celia, Myxomia series, Finesat group, Minimus subgroup, and Minimus complex. Okay, this slide shows the suggested websites invented by Dr. Laf Harbash, who is the world authority of mosquito taxonomy. Yeah, if you want to know about the mosquito taxonomy, you can use this website for the reference. Yeah. You can see the list of various species from this website. And the question for morphological identification. As I mentioned earlier in the former slides, that Anopheles species complexes difficult to separate by morphology of adults and or larvae. So we always add sensulato. Sensulato means the broad scent. You can use both italic or upright for sensulato or complex. For example, Anopheles diala sensulato, Babylotris sensulato, Corycifasi sensulato, Minimus sensulato, or Fubiatilis sensulato. Or you can use complex when you write this Anopheles species complex. This is show the example of the misspelling or incorrect species. For example, this is the World Malaria Report in 2020. If you see from the report, Anopheles albibanus does not occur in Asia. Yeah. This species occur in Central America or Africa. And Anopheles babylotris, we have to use complex or we have to add sensulato. Yeah. Anopheles fubiatilis is a species complex, so add sensulato after the name or the species of this mosquito. Anopheles lateri is a misspelling. The correct one is Anopheles lateri. Anopheles punctulatus sensulato is not species complex. Is Anopheles punctulatus group. Yeah. For Anopheles sinensis, is not species complex, or we cannot add sensulato after this species. It should be Anopheles hycanate group. And Anopheles yasuchiroides is the synonym of Anopheles paulus. So we have to change to Anopheles paulus. This is the example that why the correct identification of mosquito is very important. And if we like to search for a new species or discover a new species, yeah, what are the possibilities? Yeah, I think our audience in Indonesia, you can discover new species. Yeah. To date, many genetic species of sibling species complex await formal names or discovery. Many subspecies and variant forms identify morphologically, genetically, or molecularly, await further investigation for their specific status. And a large number of species lack description of adult or immature stage. Many species do not have a certain taxonomic position in the classification. And moreover, many unknown species are expected to occur in many areas, especially in isolated and unique ecosystem like high mountains and islands where the survey have never been conducted or have not been thoroughly conducted. So I think, however, now there is a critical shortage of mosquito taxonomists because many senior mosquito taxonomists have retired or passed away, while the number of young researchers who are interested in this field has dramatically decreased. So, are there any students or participants in the class interested in studying mosquito taxonomy?
Yes, of course, Dr. Atiporn, we have some students that working in species taxonomy. So,
oh wow, very good. Yeah, so
yeah,
they are the destiny. They are the, I think next step they are good are the excellent taxonomists because in Thailand also we lack of the taxonomists. In our department, the taxonomist is Professor Patya. Yeah, now he already retired. Yeah, so we waiting for the new generation or the young researcher, who like interested in study in mosquito taxonomy. And we several years ago we have one PhD student from Indonesia, Dr. Dia. Yeah, she studied on Anopheles maculatus in Indonesia. Yeah, and she is the advisee of Professor Patya. And I will talk about the interesting species in next slide later.
Yeah, I think Dr. Rusdia also here in attending this meeting in this lecture. Hello,
in the
Dr.
lecture.
Rusdia. Yeah.
Dr. Dia. Wow. Yeah.
Hello, Ajanoy. Nice to meet you in the Zoom.
Oh, nice to meet you. Yeah. Oh,
Yeah, very happy to meet us together.
Yeah, I hope to see you in Indonesia in the near future, Dr. Dia and Dr. Triwibowo and everyone in Indonesia. Yeah. Okay. Yeah. And how to describe and name a new species? Yeah. We follow the rule of International Code of Zoological Nomenclature and binomial the combination of the two names, the first being a generic name and the second a specific name that together constitute the scientific name of a species. For example, Anopheles genus, Anopheles subgenus, Anopheles Saeung, and the name of describe my student Dr. Taai and Dr. Habach in 2015. And the specific name must be unique within the genus. And also, the name must be published in a publicly accessible journal. We have to describe the morphological characters of adults and all immature states. And designate what the holotype is, etymology, the type series, the holotype and paratype, describe the type locality of the specimen correct systematics, and repository of the type specimen, bionomics and distribution, and etc. This is example of how to describe a new species. For example, Anopheles Saeung, species nova. we have to explain the detail of the number of the male or the female collect, the stage of the mosquito, and every and all study regarding the new species, and what techniques that used for studying this species. For example, methylphase karyotype or molecular analysis. And I will talk about methods to identify the mosquito. In the past, most species were described primarily based on morphological identification, cytogenetic, protein electrophoresis, and cross-matching methods were popularly applied to help the recognition of sibling species for a new decade. These methods, however, have several limitations. Therefore, these methods have received less attention after the variety of DNA methods or molecular methods. For example, DNA sequencing and allele-specific PCR, multiplex PCR, phylogenetic analysis, and now some researchers apply the molecular protein. For example, they use the multi-top mass spectral for identify the species by compare the protein profiling. The molecular technique have proved very useful in helping to identify mosquito species accurately, which is essential for not only the surveillance and control of vector transmitting pathogen to human and animal, but also the understanding of mosquito diversity and fauna. And I think you may know about the DNA barcoding because this is one of the molecular approaches popularly used to obtain taxonomic information from the unknown specimens. We call DNA barcoding. The method initially involved sequencing a short fragment of the mitochondrial DNA, cytochrome oxidase subunit 1 or CO1 or COX1 gene. We call DNA barcodes, and performing comparison with a library of DNA database of known taxonomy as a reference. And in Thailand, we use the mosquito dichotomous keys, which is a simple to use identification guide for the non-taxonomist by Dr. Ratana Ritikun and Arthur from 2005 to 2010. However, members of the species complexes may differ in several ways, including biting behavior, vector capacity, distribution, and insecticide resistance. The members of species complexes are usually indistinguishable or difficult to identify with certainty by traditional morphological methods due to their similarity or overlapping characters. In addition, morphological identification can also be problematic when trying to identify young instar, eggs, and damaged specimen. Yeah, it's very difficult or is a problem for correct identification. And the techniques used in the recognition of sibling species member in complexes So far, at least one and two traditional techniques have been used widely for the recognition of shipping species and or subspecies members at post and pre-mating barriers. For the post-mating barriers, the hybridization or cross-mating experiments using the artificial mating techniques to determine the hybrid non-viability, sterility, or breakdown is still a useful tool for recognizing Anopheles species complexes. And please be noted that laboratory-less colony established from naturally mixed population should be omitted since it may be a mixture of cryptic species. For cross-mating experiments, we have to question this point. And here are the details of genetic incompatibility criteria, including low or lack of insemination, embryonation, hatchability, larval survival, pupation, and emergence, and adult sex distortion, abnormal reproductive system, and complete or incomplete asynaptic salivary gland breathing chromosome. And how about the artificial forced mating? Have you ever had an experience to do this experiment, to do the forced mating?
Yes, in our lab in Salatiga is also do the post mating also.
Post mating.
Yeah, for the other lab maybe from.
Okay, so some audience have an experience. Yeah.
Yep.
Yeah, and for the pre-mating barrier. As for pre-mating barrier, the electrophoretic variation at the enzyme loci are not only useful for identification of shipping species, but also for the correct identification of morphological cryptic Anopheles species. But nowadays, this technique is rare to use. This technique detects the degree of absence in heterozygote, or we call the band mob. However, at least two problems have been raised regarding this technique. The first one is the specimens must be fresh or frozen until analysis, and it requires a large amount of sample materials to perform Hardy-Weber equilibrium and cannot be performed in allopathic Anopheline populations. For the other supplementary techniques, classical markers or the variants and molecular markers, morphological markers cannot be applied to some species, such as those in the Dairas complex that have identical or minimal distinction. Yeah. For example, this is the figure of the abdominal pupa theta of theta 9 fourth. Yeah. We cannot differentiate among three species within Anopheles Dairas complex that are Anopheles Dairas sensucitro, Anopheles scaloni, and biamii. They have the short, theta, pupa theta, whereas Anopheles carescent have a long theta with the barb. So this technique cannot be applied for this group. We have to search for other techniques that can be applied for this complex. And in 2017. This is my research that we have reached in parasite and vector. We apply to use the using scanning electron microscope for studying and identifying sibling species member in the minimus complex. Yeah, and we found that the last sensilla siloconica, this is the antennal sensilla on the antennae of the female mosquito. Yeah. We found two types of the last sensilla siloconica. The short form, which have a peg not reaching the orifice of the pit of Anopheles minimus, and the long form, peg extending beyond the orifice of the pit. Yeah. We classify it as the long form of Anopheles harrisoni. So we suggested that this technique, scanning electron microscopy, can be used for identifying two species of the Anopheles minimus complex. So if anyone like to identify or like to study other complex or other group of Anopheles in Indonesia, you can apply this, try to apply this technique for identifying the species complex. Likewise, a cytological marker cannot be applied to some species, such as those in species that have identical or polymorphism metaphase chromosomes, like in Anopheles barbilotris. We cannot use cytological marker for Anopheles barbilotris complex and Anopheles sinensis. Only for Anopheles minimus that have a distinct metaphase chromosome. So we have to search for the other techniques or other methods to identify this group. Regarding the modernized techniques, the molecular investigation of some specific genomic markers, for example, the ribosomal DNA, we select some specific genomic regions like IGS or internal transcript spacer 1 and 2 or the third domain, or mitochondrial DNA, CO1, COX1, CO2, cytochrome B, ND1 or ND5, have been used extensively as a tool to characterize and or diagnose cryptic members in the intrataxa of Anopheles mosquito. The advantage of this technique, only a few nanograms of DNA from preserved specimens required for PCR. Very popular technique for studying Anopheles species complexes. However, controversy arose when only comparative DNA sequence analysis of some specific genomic regions were used as a first-hand criteria of differentiate between the status of specific species or sibling species and subspecies within the taxon of Anopheles. For example, based on a comparison of the third domain of 20S of ribosomal DNA, Anopheles fluviatilis S has been considered as synonymous to Anopheles harrisoni. However, subsequent investigation of the conspecificity of these two species based on ITS2 and D2 and DT domain of 20S, Anopheles fluviatilis S and Anopheles harrisoni do not deserve to be synonymous. So this is very important to select the specific region. If we use, for example, if we use CO1 or CO2, but it's not it's unsuccessful, we have to try or select more specific regions to confirm the species status of Anopheles species complex. And similar results were also obtained in determining on specific species status between Anopheles lesterii and Anopheles pallalii. Yeah. This is one of the study of our PhD student. Yeah. We would like to prove the true species status of Anopheles lesterii and Anopheles pallalii. Yeah. These two species have a distinct microhabitat, and lesterii found in fresh water, for pallalii in the brackish water. For the wing characteristic, these two species have the overlapping wing morphology. So we cannot use wing for identifying these two species. And we compare the sequence between these two species. And the result reveal the low, genetic distance for CO1 and CO2. Whereas considerable genetic distance was obtained in ITS2 regions with 16-base substitution. Supportively, the phylogenetic tree demonstrated that these two species were separated from each other with 74 to 100 bootstrap value for the three regions. But I show only one region. It was interesting to note that Anopheles lesterii and Anopheles pallalii were confirmed to be genetically compatible by the cross mating experiments. So, from our study, we suggest that the gold standard for before we conclude the species status of Anopheles species complex, the cross mating experiment is the gold standard for definite conclude the species status, because as I mentioned about the molecular techniques, even they show the two species separate from each other, but the result from cross-mating experiment reveal that the two species are the same. And next, I will I will talk about the Anopheles Species complex in Thailand, because when I was a PhD student, I studied this group in Thailand, and our team, my advisor, Professor Saeung, now he already passed away, and I and our team, study this group in Thailand, and we discover 3 new species member within Anopheles Species complex. We discover Anopheles Species complex for the first time, and we also discover 3 new sibling species in this group. And we wrote the systematic technique for the recognition of Anopheles species complexes in the chapter of the book Anopheles mosquito, New Insight into Malaria Vectors. Professor Saeung is the editor of this book. You can download my chapter or other chapter via the website. Yeah, and we used this robust systematic procedure for studying this Anopheles species complex. You can see by following the flow chart. We first we try to collect the Anopheles mosquito that are distinct in their behavior. For example, biting in humans or animal with relation to different microhabitats and or locations. And then try to record morphological variations as far as possible during the species identification process of wild-caught females. And we establish an iso female line or isoline colony by allowing gravid females to lay eggs individually. And we conduct the molecular investigation of the laid egg feral female to obtain a robust DNA marker, with this step usually taking about 1 week. Since the development of F1 progeny usually take about 2 weeks from the first instar larvae to adults, the metaphase karyotype investigation of fourth instar larvae or newly emerged adult female and males is performed in order to obtain a cytogenetic marker. If molecular investigation fails in the step of lay egg feral female, it will be performed in F1 progeny. And we also carry out the morphometric and morphological investigation of the egg, larvae, the pupa, skin, and adults to confirm a precise species identification, and perform the step of cross-mating experiment among the iso female line colony by using karyotypic marker related to the DNA marker, which have a large sequence divergence or very low intraspecific sequence variation of each iso female line colony. And this is the technique necessary for the establishment of difficult to rare Anopheles species, which yield the high rates of attractive metaphase and protein chromosome and the potent adults for cross-mating experiments. We use slightly more or less Malaysian grass, 15 stem of the garden grass, depending on the size of the stems and the size and number of the leaf, provide to improve condition to a suitable level for larval rearing. Since the glass provide a resting place for larvae, lender shape as in natural breeding sites, for example, the rice paddy, the ponds, and the swamp associated with the water plants, and aerated the medium. And the roots of glass were also very important for maintaining clear and clean rearing medium by using larval waste products and unconsumed food as fertilizer, which determine the obvious active growth of glass in the rearing tray. Yes, this you can see our technique for rearing the mosquito in the insectary. Yeah, and this is the technique that we use for preparing the metaphase chromosome. And the colchicine solution has been used widely for metaphase chromosome population in the cytology study of eukaryotic organisms. For example, the protozoa, the helminth, the snail, insects, and plants. The spindle formation of microtubule polymerization inhibits arresting mitosis at the metaphase. We inoculate of 0.5% solution of the dry decorative plant Gloriosa superba seed powder into the post spilacular area of the mesothorax, and a filtrate of solution was introduced into each mosquito by gently blowing down the attached rubber tube. Here is the figure of our metaphase chromosome derived from ovary and the testes of the adult female mosquito of Anopheles pallari. Yeah, this is the flow chart to prepare the dry Gloriosa seed or rhizome powder. Yeah, as you can see in this slide. Yeah. By keeping at this condition, the colchicine-like activity in the filtrate could be stable for at least 2 years, and we can save costs for no need to buy the synthetic colchicine from import from abroad. We can prepare by ourselves by using this decorative plant. In Thailand, we call this, the name of this plant is dong dung. It has a colchicine-like activity. For cross-mating experiment, this technique were conducted among the laboratory-raised isolines. The adult F1 hybrids show sex distortion, abnormal development of ovarian follicle, and atrophy of accessory gland and testes. You can see in the figure, this is the control of the normal ovary and the testes. If we test we cross some unknown species between A and B, and we check the result, check the genetic incompatibility, compare with the control. If we see the abnormal ovarian or atrophy of the testes, it means that this species have a genetic incompatibility. Yeah, and as I told you that I study Anopheles species complex since 2005, and 10 years later, we described the 3 new species of the Barbarossa complex. This is my student and Dr. Habash. We described 3 new species, Anopheles dissidens, Anopheles saeung, and Anopheles wechushotai. We use the name to recall to Professor Chushot, who is the first person to study this group in Thailand. And I'm deeply honored that Dr. Lav also give the name of this new species, Anopheles saeung, for my surname. This is the phylogenetic tree of Anopheles Barbarossa complex. You can see this is Anopheles van der Waals, collect in Sumatra, Anopheles van der Waals, and we compare our sequence with Anopheles van der Waals and Anopheles barbarossa, and some sequence published by other researchers who collect Anopheles barbarotis from Sumatra. Yeah. Anopheles Saeung. So after we compare the sequence of both ITS2, CO1, and CO2, yeah, we found 3 new species. Actually, when I was a PhD student, I use only species A1, A2, and A3 because based on the cytological marker of the metaphase chromosome. And after we describe the new species, we change species A1 to dissident, species A2 Anopheles Saeung, and for species A3, we last year we have study insight into this species again because we want to know the true species status of this barbarotis species A3 in Thailand. We try to collect a mosquito and re-identify their morphology, larval stage, pupa stage, and we found that Anopheles species A3, the true species status is Anopheles hodgkini. So no barbarotis species A3 anymore. We already published in 2023 in last year. Yeah. And for Anopheles completed life form E, we name it as Anopheles visual eye. And this year, our team, Professor Pratya and his colleague in Bhutan, we have published and we discover a new species, Anopheles sapangensis, of the Anopheles barbarotis complex. We just this paper we published the identity of Anopheles barbarotis species A3 of the barbarotis complex last year, and we found that is Anopheles hodgkini. And this year we published the new species Anopheles sapangensis. This species described from Bhutan. Yeah, and I like to give an example of Anopheles maculatus group by Dr. Ari and our team in 2019 when Dr. Dia is our PhD student. Dr. Dia published 2 papers. Her PhD thesis is about the study of the genetic of Anopheles maculatus group in Thailand and in Indonesia. And we published the paper, Genetic and Morphological Evidence for a New Species of the maculatus group of Anopheles subgenus in Java, Indonesia. So Dr. Dia can add more some information, some detail for us as well. Yeah, we use the cytological marker to study Anopheles minimus group in Indonesia, and the metaphase chromosome of the Javanese form. We found the acrocentric and telocentric form of metaphase chromosome. But for Thailand, the X and Y chromosome is the last submetacentric chromosome. For the larvae, the city of maculatus larvae from Java, figure A, 2D, you can see this larvae from Java. And from figure E, 2H is the maculatus larvae from Thailand. Yeah, you can see the different morphology of the city of maculatus larvae. And also you can see the morphology of the abdominal theta 9/4 of the pupa of Maculatus sensucito Thailand strain in figure A. For figure B and C are the abdominal theta 9/4 of the pupa of Maculatus Java strain. And here is the abdominal theta 9/5 of the pupa of Maculatus Java strain, which have the different morphology. Yeah, and also we compare the theta 2C dorsomentum of the head of the larvae and found the difference between the Java strain and Thailand strain. For the crossing and back crossing experiments, this is the cross between the parental. You can see we can compare the number of eggs per oviposition, compare the percent embryonation and percent egg hatch with the parental strain. We found the low percent embryonation and percent egg hatch, percent immersions, and percent female and male with abnormal ovary and testes, respectively. And when we check the reproductive organs of the F1 hybrid, the life form crosses between Anopheles maculatus Java strain and Anopheles maculatus Thailand strain. You can see the abnormal and the sterile testes of the reproductive organ. And the testes of hybrid male derived from backcloth, showing the abnormal spermatozoa with a large head. So from the result of the cross-mating experiment, it means that the Anopheles maculatus Java strain and Thailand strain reveal the genetic incompatibility. And also the supportive result from the molecular analysis reveal the true trait of Javanese form and Thailand form, Thailand strain of Anopheles maculatus. So this is very interesting to study this Anopheles group in Indonesia. To maybe you can describe the new species of this group in Indonesia. And I like to add some information about the climate change and adaptations. What is the climate change? Yeah. What is the climate change from the audience? Could you give me some information about the climate change? You can write Thai in the chat.
Yes, maybe there any person that will answer the question. Maybe you can answer also in the chat box.
Yeah. What is the climate change?
Or maybe someone would like to answer directly. Please
Answer
raise your
directly.
hand. Yeah. Anyone?
Raise your hand. Yeah,
Oh, no. I think in the chat, I think someone.
The chat.
Yes. The chat.
The chat. I cannot see. Maybe my computer have some problem. I cannot see the chat. So could you answer directly?
Maybe Pak Arti, could you please to answer directly?
Yeah.
Please open your mic. Go ahead.
Please, yeah, feel free to answer.
Yes, thank you, Prof. Atiporn. I want to ask one question. Since the identifications process is often research intensive and complex, are there specific indicator or critical moment in the field that might suggest a mosquito species has been misidentified like that? Specifically, how can we detect such misidentifications?
Misidentification. And yeah, thank you for your answer. Yeah, the climate change refer to the long-term alterations in temperature, precipitation patterns, and other climatic variables that arise from human activities, particularly the release of greenhouse gas into the atmosphere. Yeah, change with ecosystem have the potential to significantly impact that may have noteworthy consequence influencing the distribution and behavior of various organisms, including vectors. Climate change has significant implication for transmission and spread of vector-borne disease because it impacts the biogeography and ecology of insect vectors. And the effect of climate change, the development and survival rates of vectors are directly impacted by increasing temperature, which in the turn affect their population dynamics and geographic distribution. Because of higher temperatures, the life cycle of arthropod vectors progress faster, which means that more diseases are being spread. And this slide shows the strategies for controlling vector-borne disease in climate change. First is the several primary approaches to controlling arthropods that transmit vector-borne infections are as follows. Enhance the vector control to prevent vector-borne infectious disease in the context of climate change. Enhance the effective strategy include integrated vector management, intersectoral collaboration, vector surveillance, and environmental monitoring. Public policy, strengthening public policy designed by integrating vector surveillance and control with environmental management is crucial. For environmental management, we implementing sustainable land use practice, enhancing water resource management, and for example, for the larval control strategies, such as environmental management and the use of larvicides are also employed to target mosquito breeding sites. For personal protection, since there are few vaccines available to protect against vector-borne disease, avoiding insect bite is the best method to prevent them. This can be accomplished by using insect repellents, donning protective clothing, and taking other preventative measures against insect bites. And also, we use the insecticide-treated bed nets or ITNs and indoor residual spraying or IRS are widely used in malaria endemic areas to reduce mosquito populations and prevent bites. And also education and awareness. Public health requires raising awareness about vector-borne disease and drug prevention methods. This include the education communities on the significance of private environmental management to prevent arthropod reproduction. And the last one, research and innovation. Developing, we we will develop new pesticide formulations, novel tools for vector control can help advance the prevention and control of vector-borne disease. Additional understanding the impact of climate change on vector-borne disease can help inform public health research and policy. Okay. And successful development of the vaccine and the novel medication for vector-borne infectious disease might reduce the importance of vector control. Yeah, this is the reference that I use for talking today. Okay, that brings me to the end of my lecture today. Yeah, thank you for your attention. If you have any questions or comment, please feel free to do so.
Okay, thank you very much, Dr. Atiporn, for the very interesting and insightful presentation today. So now I think we have some time for question and answer. Maybe if you have a question, please raise your hand or maybe type in the chat box. So please, this is a good time for us to discuss about the species complex with one of the most influenced expert in the world. Okay,
Yeah.
Go ahead. If you have any question, please raise your hand.
Yeah, or maybe if Dr. Dia, can you add more information on the progress of your study of Anopheles maculatus in Indonesia? Yeah, you can share your information to our audience.
Yes, please, Dr. Dia, maybe you have some additional information for this lecture.
Yeah.
Please. Hello. Okay. While we waiting from Dr. Dia, maybe one question from the chat box. What are the morphological methods used to identify mosquito from the S2? And then the morphological method relies on the mosquito key for identification. So what are the advantage and disadvantage of the approach? Yes.
Of
Can
the
you, yeah,
morphological technique, right?
Yes.
Yeah. If because as I told you earlier that Anopheles species complex, they have minimal or nearly identical morphology. So we have to use like a systematic or multidisciplinary approaches for identifying Anopheles species complexes. We cannot use only one or two techniques. Yeah. We especially morphology, we have to find or search for more techniques like cytology or cross-matching experiment or DNA analysis to integrate all technique to identify Anopheles species complex, because very difficult to identify based on only morphology. But it's very difficult. Yeah, the limitation is very difficult to establish the isofemale line or isoline colony in the laboratory. Yeah. Because for my opinion, I think the cross-mating experiment is the best technique or the gold standard technique that can conclude that this species is the species complex or distinct species or not. Yeah. So if you cannot establish isoline colony, we can use morphological techniques and DNA analysis to support together and compare your sequence with the reference sequence in the GenBank, and using the suitable genomic markers like barcoding, COX1, COX2, and ITS2. We can try to use several regions for studying the Anopheles species complex.
Okay, thank you, Dr. Atiporn. Maybe we still have another question. Maybe please, Anis, open your mic. Maybe you can ask the question to Dr. Atiporn directly.
Thank you, Mas Tribowo.
Okay.
Good afternoon, Prof. Atiporn. Thank you for the insightful presentation.
Yeah,
I'm interested in Anopheles Species Complexes in Indonesia. I'm studying under supervision of Mr. Tribowo. And my question is, which the techniques the most effective to identify the Anopheles complex, and or those techniques are complementing of one to each other? Thank you. Because actually I use 3 genetic markers, CO1, CO2, and also ITS2. And also maybe if we have more time and opportunity, we will have more for metric and geometric also. Thank you.
Yeah, I think you can use genetic marker. You can integrate or combine several regions, and because some species, if you use only one marker, we cannot conclude the species status. So you use COX1, COX2, and ITS2 is very good for studying. And if you have time, you can apply the scanning electron microscope or morphometric using now wing morphometric now have been used widely as well for mosquito and for insect. I apply the wing morphometric for study mosquito and blackfly as well. Now I think this tool is maybe the alternative method, and also the scanning electron microscopy. And if you can conduct the cross mating experiment, it may be very it may be perfect for your study. But if you use only molecular marker and have another technique to support, it maybe is okay for studying Anopheles species complex. I encourage you and hope you successful in your study.
Thank you, Prof. Atiporn and Mas Tribowo.
Okay, thank you, Anis.
Thank you so much.
Very nice question.
For your questions.
Okay, maybe a question from the Mutia. What is the biggest threats of the climate change for the malaria vectors? What the biggest threats of the climate change for the malaria vector? I read from the chat box the question from the Mutia. Yeah.
The big
threats. Yes.
Okay, I will example. Sorry, I will try to see my chat. I'm not sure. It did appear from the tool because my staff helped me.
Okay,
I will try.
Maybe the meaning of that Ajanoi is the biggest impact.
yes,
The biggest impact of that.
Oh, the biggest impact. Okay.
Yes.
The biggest impact is for the climate change, because as I told you that we cannot control or eradicate the mosquito, because the higher temperature can affect the life cycle of the mosquito. So if they have a faster life cycle or short life cycle, it means that the malaria can spread and cannot control or eradicate. So this is our the
Okay. Yes. So maybe the last question from the chat box. I think from the Nurfianto. Could you please to open your mic, maybe, Nurfianto? Hello. So, hello, Pak Nurfianto. Can you hear me?
yeah,
Can
yeah,
you
I
can
can
you?
I hear your
Yeah.
voice.
Okay. So, Nurfianto asked about the alternative plant extractor. I think this is different with our topic, but maybe if you any answer for this, you can go ahead. But if not, it's no problem. Are the alternative plant extract other than Gloriosina superba that can be used in a similar study and the potential of plant extract as bioinsecticides? So I think this topic is about the bioinsecticide. So maybe it's out of the topic. So maybe do you have experience little bit maybe about the bioinsecticide, Dr. Atiporn?
Insecticide, yeah, we have to we have an experience to study insecticide. Yeah, in Thailand, we use the a test with the pyrethroid. Yeah, and I found that only bendiocarp. Yeah, for my study, only Anopheles minimus and maculatus. Yeah, have how to say the insecticide have a low mortality of the have a low survival rate with the bendiocarp, but for other insecticide is no. So only bendiocarp. I have only one experience, only one paper.
Okay, thank you, Dr. Atiporn. So maybe the other question is there an effect of sex pheromones on Anopheles mating so that disrupts mating technology using synthetic chemical traps can be used?
You mean?
You mean this is mean that effect of the sex pheromone in related with the using this sex pheromone from the Anopheles for the chemical trap, or maybe for the mosquito trap like this.
Yeah, I think if we try to develop the chemical that relate to, I think if we develop the some chemical can, how to say, the interrupt the function of antennae. We can control the Anopheles mosquito in the field if we can develop some chemical that interrupt the function of antennae that used for seeking the host.
Okay, thank you.
As I study the sensilla on the antennae, I found many type of the sensilla antenna on the female antennae. Yeah, and we hope that we can further investigation for developing the new technology or new tool for controlling Anopheles.
Okay, thank you, Dr. Atiporn. I think all of the questions from the chat box already answered from you. So
about Dr. Dia.
Yeah, maybe do you want to add something, Dr. Dia? to add information from
Anopheles,
Dr.
yeah,
Atiporn?
that group is very interesting group, yeah, to start for studying.
Thank you, Dr. Atiporn, for the kindness. But I think until now I don't have any update information about Anopheles maculatus. That's last time I think Mas Tri doing research also about the maculatus in Indonesia. So
Yeah.
until now we don't have the new name, right, Mas Tri?
Yes, exactly. So I think we discussed maybe last year with Professor Pradia also, and also Professor Sylvie, and maybe we are together to continue maybe with Dr. Rosdia to give the name. I think it will be important to give the new name and then to publish it soon. Yeah.
Yes.
Maybe after this we can discuss together to finishing all of this our work. Yeah. And I think Dr. Atiporn, I think really support for this our work. Yeah. And actually we also have some good finding during the recently, because of we also have some very interesting information about the other species complex in Indonesia, especially for the part of the Anopheles Leptospirus group that maybe it's very complicated, but I think we got some look like unidentified mosquito that we need to clarify also.
Oh, yeah. We can, for taxonomists, yeah, you can contact to Professor Pratya, yeah, and if you need the help for identify, we can cover it together, or you can visit our department.
Yes,
of course.
Yes, of course.
Yeah, we
of course.
welcome you all, you to our department in Chiang University.
Yeah, and maybe Ajanoy, you can share your protocol about the cytogenomic method, but because last time we have student want to study about the cytogenetic, but I think they don't have any protocol about how to do the metaphase chromosome. So maybe you can share the protocol about that. And then I think the one question, because in Thailand we use dongdeng, right? Dongdeng to change for the colchicine. So maybe you have any suggestion what kind of the material we can we use for change the dongdeng, because in Indonesia we don't have dongdeng.
Yeah, you can buy or purchase the synthetic colchicine. Yeah, you can buy it. Yeah, but we try to like save the money. We stop, and try to use the this plant, and we found that it have a effective activity like the synthetic colchicine. Yeah, you can buy, or you can search in the market. I think you can buy the synthetic solution of colchicine active activity.
Thank you for your suggestion.
Okay, and I can share the protocol for metaphase chromosome preparation to you and your student via email.
Thank you.
Okay, thank you very much. If there is no more questions, so I would like to thank you very much to Professor Atiporn Saeung for your insightful presentation today. I think this is very useful for us, especially for the all people that working in a mosquito taxonomy. And of course, I will like to also thank you very much for all of the participants, especially here is from the Ministry of Health, and then Mbak Dia also from the brain colleagues, and also I think Dr. William Howley also present, also attend in this meetings, this lecture. Thank you very much also Pak Phil for your attending here, and thank you very much. We all really appreciate your presence today, Professor Atiporn, and for all. Look forward to seeing you all our next lecture. So again, thank you very much, Professor Atiporn Saeung, for the very nice presentation, and very happy having you. Yeah.
Yeah, I'm very grateful for your kind invitation. Yeah, I'm very happy to talk to you all here today. Yeah, I'm deeply honored to be invited to speak today, and hopefully our audience can get the information or some idea for your studying. Yeah, and hopefully that I can have a chance to talk to you all again in the near future. Thank you so much for your invitation.
Okay, thank you very much, Dr. Professor Atiporn.
Okay,
So thank you all, and yeah, happy to see you, and have a wonderful day. Okay, thank you for all, and especially for Professor Atiporn. Yeah.
Thank you so much. Okay, ขอบคุณค่ะ
Okay, ขอบคุณ
Bye
ครับ
bye. Have a
Bye
nice
bye.
day
Yeah.
ahead. Bye bye.
Okay, thank you.
Thank you.
Oke, terima kasih kepada Bapak Ibu semua, dan barangkali perkuliahan tamu siang hari ini bisa kita tutup bersama. Kami persilakan kepada tim dari Tropmed untuk bisa teman-teman dari Tropmed untuk menutup Zoom ini. Ya, terima kasih. Assalamualaikum warahmatullahi wabarakatuh. Ya, oke, bye bye everyone.
Okay, bye bye everyone.
Ya, oke. Ya. Bisa ditutup, Mas. Ya, monggo dari
Salam. Lah siap baru buat siap malah.